of really starting with like some review of the last lecture. You guys know what a capacitor is. You guys know that the membrane acts as a capacitance. Everyone's comfortable with that concept? We'll use some of these concepts to understand synaptic transmission. So capacitance, separation of charge. The larger the cell, the capacitance will be proportionately bigger, because that will be the time it will take for the membranes to charge, right? Okay, and then we know ions only flow through ion channels, so those are the conductors, and when they're closed, they act as resistors, right? Then we have potential difference that gets created because there is separation of charge, and so we have a potential difference or a voltage. All right, so I think this is all basic. You guys are comfortable with this, right? This is what an action potential looks like, and the underlying conductances. Anyone can tell me why? So what is ENA? Reversal potential of sodium. What does that mean? Quickly. Yeah. So at reversal potential, what is going to happen? The movement of the ion stop. Why? Is it voltage? The movement of the ions. There's no net movement of the ions, right? Okay. So why doesn't the action potential reaches the equilibrium potential of sodium? Why doesn't that happen? Three main reasons. So till it reaches the reversal, the concentration outside will always be more than inside, right? Okay. Okay. So that's happening, and that happens when it goes closer. It's going to stop only when it reaches to the ENA, right? But it never reaches there. So you are right that there is a net positive charge that goes in, and it acts as a repulsive force. So there are two forces that result in an equilibrium potential. One is your driving force. One is your electrotonic force, which is your if it's a charged particle, it's going to repulse the further charged particle that's coming inside, right? And that determines your equilibrium potential. But why in this case, even the sodium channels are like, it never reaches, the peak never reaches the equilibrium of sodium. Okay. First thing is, as you continue to depolarize, your sodium channels are very fast to open and very fast to close. So they don't close. There's another term. They inactivate, right? So the sodium channels start to inactivate. That's number one. Second, potassium channels start to open up. So now they are driving the potential to the other direction. Okay. And third, what is the third? Is there any third? I mean, the third is that it is going closer to the, so the driving force itself is reducing. So even if you have the sodium channels open up, the rate of movement of your ions reduces because you're now closer to your reversal potential. Okay. All right. So sodium channels, conductances, you see that it's very fast to get activated and very fast to go down. Potassium channels are much more slower. And then what happens here? So this one, you see, this almost reaches to the reversal potential of potassium, which is what? Any random number? Do you guys know? Around minus 90, right? This offshoots to minus 90 because there is no, regulatory or no other ions that are contributing to this downward movement of your potential, right? So it literally offshoots your resting membrane potential. And then how does it come back up? So pumps is one, leak channels, chloride channels, right? Okay, cool. So these are the ion channel contributions. Just wanted to see that. And the last concept is this current voltage relationship. Are you guys comfortable with this? I always basically cover this because I feel like this is a very cool thing that Hodgkin Huxley did. This is what they made, right? This is their discovery. It's what this is. What is IV? Current and voltage. At a particular voltage, what is the current response? And so for potassium, it's kind of simple, right? You go from minus 70 or minus 80, it's kind of closed, right? Any ion movement will happen? No, yes, no. It will not happen. Why? because it's voltage gated. So it will only get activated when it's depolarized. Okay. So with potassium at -60, -70, there is no current flow. Slowly, as you keep increasing your voltage, it go from -60 to -50 to -40, you will start to have some ionic flow. And also because you guys know that the reversal potential of potassium is -90, you're always going away from the reversal potential. So your current will be almost proportional to the voltage change, because the only thing that's driving is your the concentration gradient, right? And so you're going away from the reversal potential. So you'll have a linear curve like that, right? Simple. Potassium is simple. And this is a IV curve of potassium. Now, how does the sodium curve look like this? Why would that look like this? So again, at -50, -60, all sodium channels are closed, right? As you start to open up, you don't have a very high, you don't have like um, 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 you have small amplitude because the sodium channels are opening. So in the beginning, it's kind of linear, but it peaks somewhere here, which is around -20 and -10. So what does that mean? What does this peak mean? Yeah. So all the sodium, imagine like all the sodium channels are open, and okay, and then what happens if you keep moving up? So it's not just the closing, but it's also, yeah. So now you're going closer to your reversal. So the reversal of sodium is +55, right? So your ionic flow will start to reduce, even if the ion channels are. So of course, it's a combination of both opening and closing. But even if they're open, even if you do an artificial constitutively open sodium channel, you will still have this reduction because it's now going closer to your reversal potential. So that's why reversal potential of a particular ion channel is very important. And that's what people use to study its different, like it's one of the most important feature that they use to study the ion channels. Okay, reversal potential of that. All right, so simple, simple, simple. So today we'll talk about synaptic transmission, the basics of that. Historically, we didn't know that there are many different types of synaptic transmission. Right now, we know that there are, you guys have heard about chemical, electrical. Back in those days, like 20 years before this, the neurophysiologists were still debating about whether it's a contiguous transmission or it's a continuous transmission, right? So um, um, Golgi and Ramoni Kahal, even on the day they got the Nobel Prize in 1903 or 1904, they were discussing. So Golgi used the Golgi stain, and he said that the brain has these mesh-like structures, the network-like structures, and that's how they act. So they are these isolated meshes, and that's how they function. Whereas using the same technique, Ramoni Kahal drew the neurons and say that the neuron is a independent unit of processing the information, and they are not really connected like a wire. They are connected in a contiguous fashion, such that they come close to the other neuron, but they're not connected, right? And so 20 years later, people were trying to understand what is the nature of this information transfer, whether it's electrical in nature or it's chemical. And there are these two physicists, not physicists, physicians and anatomists, Otto Louis and Henry Dale. They discovered for the first time that there could be some electrical to chemical transduction that can happen during neurotransmission. And so what they did was, I'll not use this. All right, so this is something that you guys did. So the experiment of Otto Louis and Dale, you guys have probably seen the vagus nerve stimulation. Have you guys heard about this experiment? Okay, so I'll tell you. This is something, so they isolated the heart with their nerve intact, and they put it in the Ringer solution, which is kind of like containing salts and sugars. And then what they did was they stimulated a vagus nerve here, and then they only had the fluid transfer to the other side, and they saw that the heart responded to this stimulation. This heart responded to this stimulation. So they were recording from the other heart, which was in the other. So it's isolated. They did not contain any nerve, but then it responded to this vagus nerve stimulation, suggesting that there is something in the fluid that was basically being transferred. And this was again a frog heart. so they were doing the stimulation and they can see. So vagus nerve stimulation really releases these chemicals like epinephrine or norepinephrine, depending on the frequency. And in this case, it reduced the heart rate, right? And so they saw that the one that had the intact vagus nerve, you do the stimulation and you'll see the same response in the other heart as well. So there's something in the fluid that got transferred, right? And so then later, series of experiments shows that they were able to isolate which chemicals there were, which neurotransmitters are there. And now we know we have many different types of neurotransmitters, neuromodulators, which are different from neurotransmitters. So we'll talk about that. But it started from these experiments. Today we know that there are different types of synapses. Usually, where are the synapses formed? Between which two neuronal regions, neuronal structures? Axon and dendrites. So exodendritic synapse is one of the most common ones. But there are other synapses that are being formed. Can you guys think of an example of an axosomatic? Peripheral neuromuscular, kind of, because muscle doesn't have that specialized structure. But we'll not call it as a soma of a muscle, right? We'll call it as a cell, muscle cell. But that's a valid example. What else? Touch reception. Okay. Let's see. So there are, but again, it's not between two neurons, right? So it's again maybe a touch cell, and that doesn't have a specialized structure or have axons and dendrites. So example of axosomatic could be interneurons, right? So there are a lot of these interneurons or GABAergic synapses that are made directly onto the soma. So they don't make connections like paravalvulin positive neurons are making connections on the soma itself. So this is in the central nervous system. Axoaxonic, exactly same. There are a lot of interneurons that make. So axoaxonic is not very common, but the ones that I know of are the one that make synapses near the axon initial segment. That's where the action potentials are formed. So they are mostly GABAergic synapses. And then dendrodendritic. That's also very interesting, right? So the dendrite itself has so it makes the connection with the other dendrite. So it's shunting the entire somatic and axonal part. What would be the function of this? Dendrodendritic. Exactly. So this is there, but can you guys think of like just bypassing your entire rest of the cell? To amplify, to do the to coordinate between the cells. So if you really don't want any processing or any loss of information to happen, you can really have multiple dendrites connected. So it's also there in the olfactory bulb to do the odorant processing, also in the retina. So a lot of the early sensory processing regions have these dendrodendritic structures.
So dendrodendritic connections in the OP, for example, I think there is a lot of inhibitory synapses there, and they can clean out the data that otherwise comes from the activatory synapse.
Absolutely, yeah. So exactly. So we don't cleaning out meaning like, yeah. So when you have information coming to the soma, it gets integrated with a lot of different inputs as well, and then the processing happens and then it filters out. So in this case, we kind of don't want to filter out. We kind of just have coordinated information sent to multiple cells at the same time. So even the retinal ganglion cells, the amacrine starburst cells, they show the same thing in the retina. Again, very early during the processing of sensory information. So those are some of the anatomical types of looking into the synapses. When we just take a snapshot and look into the synapses, we'll see some examples of that. And then you guys know about the physiological types, right? Chemical and electrical. Today we'll mostly focus on the chemical synapse, but I'll just quickly go over the conjoint. So conjoint synapse can have both electrical and chemical. So that also exists in a lot of the mammalian systems where you do have electrical connections with the help of connexins, but the main source of your information transfer is still chemical at the end. Actually, no, that's not. So a lot of the synapses, at least in the lower order invertebrate models, are electrical in nature. They're quite fast. They don't have directionality, and not a lot of processing takes place, or the processing in the sense, not a lot of computation takes place when it comes to information transfer from one cell to the other. Yeah. Most of the other brain region. Actually, most of the mammalian systems, um, um, like you are processing any, so pain information, for example, you have a pain information that comes in from your skin cells, and then it gets transferred to your brainstem region in the beginning. That's purely chemical. There's no electrical. Um, it's not just slow. Even the lot of your even your muscle twitch, everything is chemical in nature, right? So your muscle movement, you want to, so the neuromuscular junctions are all chemical in nature. You can have a lot more computation. Like, so there are a lot of these processes, like a lot more complex processes are involved, which you can control in a very regulated manner. For example, the amount of neurotransmitter that gets released, the amount of receptors that are there to receive the neurotransmitters, the amount of action potentials that can come down, and or the amount of vesicles that are there, which we'll see today. But none of those components are there in the electrical. So electrical synapse is mostly like just the information comes and passes through. So it's almost like a tunnel. Whereas in this case, you have like a check post to check at that level as well, and then move forward. That's the difference. We know for sure. I don't know that. So, yeah.
Did we move towards chemical synapses because our peripheral kind of nervous system, you're moving towards the center?
Yeah, it's quite possible that evolutionary speaking, we started to get more chemical or more specialized synaptic structures, and it was not just, connecting based movement of ions. The and it's like always a energy cost, benefit thing that you have to see, because it's also energetically expensive to maintain these chemical synapses as compared to the electrical ones. But I don't know. When did it start to appear? I think most they were. It's just that the proportion of the chemical synapses increased a lot more than the electrical ones. All right. So yeah, simple. So this cartoon is just to describe the same thing that I was telling you, that for the chemical synapse you'll have a lot more components which will allow you to regulate the information that is being transferred. You have synaptic vesicles. We'll talk about these. Whereas the electrical synapse have these gap junctions, right? So the ionic flow can happen through these gap junctions bidirectionally. So if the action potential is generated here, it can go to the cell upstream. If the action potential is generated in the upstream neurons, it's going to go travel downstream. So there's a bidirectional communication between the neurons. So this also tells you that you kind of don't need specialized structures like dendrites and axons and these kind of structures. Like you can have something which is like unipolar neuron, and you can still have that communication. But today we'll focus on the chemical synapse. So I'll talk about how did we start to study the properties of chemical synapse, and then where we are now. Mhm. Absolutely. So we'll come to that. So in addition to the connection that is just happening at the level of synapses, you can have hormones or neuromodulators that can be released. It'll still be released from the axonal boutons or axonal terminals, but it'll be released in a volume fashion. So it's not really having a very specific postsynaptic partner. It's going to basically be received by many synapses or many cells at the same time. So if it gets released around the soma type, I think this is called volume transmission. So the release is happening by many axons at the same time, and many soma or many dendrites get the information. But this is what's known, but now we know that even that process, even this volume transmission is happening at a in a very regulated manner, right? So there are a lot of these roles of non-neuronal cell types like astrocytes and interneurons that can regulate the coordination of the release of neuromodulators, even if it's volume. So it's a lot more control now. Okay. So basic properties of a synapse. Can you guys like this cartoon is something that you guys have all seen, right? This is very simple cartoon. What is this kind of synapse? What is the cartoon of um as in this cartoon is inspired by which kind of synapse? It's a chemical synapse, of course, but any particular kind of chemical synapse that you guys can think of? Calcium gated, they all are calcium gated. It's actually not acetylcholine. This structure tells you something specific. It's a spine, yeah. So that's how we have started drawing a synapse, but this is not really how all synapses look like. This is an example of an excitatory synapse, which are made on these specialized structures called as dendritic spines. Anyways, so there are three main components of synapse. One is you will always find a synaptic vesicle. Okay, you will always have the postsynaptic receptors, which will receive your neurotransmitters, and then you'll have something called as a synaptic cleft, which is a space between your pre and the postsynaptic partner. And this darkened portion that I see again is a characteristic of a excitatory synapse. So I'll show you the real schematic of how a chemical synapse look like. So usually neuroscientists are not very, they don't imagine, these things. They usually like to see things, right? So whatever they see, they kind of draw it. And so this is something that they actually see it. So this is an electron micrograph where this is a dendrite. It's coming out like this. Okay? Soma is somewhere far behind. And so we have made just thin sections where you can see dendrite. This is the dendritic spine, and this is the presynaptic partner. Okay? Are you guys all seeing this? What you see here are the synaptic vesicles. What you see in between is a synaptic cleft, and then there is a darkened portion. Can anyone tell what that darkened portion is for an excitatory synapse like this? It's the active zone, absolutely. What does that active zone contain? So what's the darkened portion? Why is that active zone dark? With the help of, so channels are there, but the darkened portion is not because of the channels, because of the proteins with which these channels get anchored. So it's this lot of scaffolding proteins like postsynaptic density, PSG95, and a combination of those. So there's a high concentration of proteins there. So that's why this darkened portion, that's actually called the active sites. That's absolutely right. Yes. Is there an active zone in the presynapse? As far as I know, there is a. So yeah, so the active zone, there is also an active zone, but that's something that you don't really see visually in an electron micrograph. So the active zone is something that physiologically you determine by seeing where the readily releasable pools are, where the dock vesicles are. But anyway, so this is an example. What else? What else interesting that you see here? What are these black things? Mitochondria. What are these? What are these small little dots that you see in the dendrite region? What are those? Hmm? Yeah, I think yeah, microtubules exactly. So those are microtubules. What are microtubules? They are an example of a cytoskeleton protein, right? So what is the function of a cytoskeleton protein? Provide structural support. Is that it? What else? Absolutely. So nucleus is somewhere like 1 millimeter away. Dendrite is somewhere there to really, have translation happen or have any kind of communication happen, there are a lot of these motor proteins or cytoskeletal proteins that play a role in communicating between synapses and the nucleus. Is there anything else important? How many synapses can you guys see? Three. So yeah, very easily you can tell by, so see, there are some other synapses that are being made directly onto the dendrite. So not all synapses are made on the spine. So 1, 2, 3, which is the fourth one? Where? Oh yeah, yeah. So the 1, 2, 3, 4. Yeah. Which one? This one? Yeah. Interesting, right? So I don't know. Yeah, it does look like a presynaptic component. Okay, cool. So this is an example of a, but that's how you start visualizing these synapses in real, right? And the cartoon that I showed in the previous slide was very similar to what they actually observed. Now, they observe different types of synapses. So there are inhibitory synapses and excitatory synapses. I'm just dividing them into two broad categories. And this is again an electron micrograph where I kind of want you guys to know the difference between an excitatory synapse and an inhibitory synapse. Can you guys think of some differences? Let me see. So I is an inhibitory synapse and E is an excitatory synapse. Can you spot like three main differences? It's darker. Okay, good. That's actually true. It's very true. So the active zone, the darker and active zone is actually a characteristic of an excitatory synapse. Okay, what else? So that's one. The other could be simple. So the inhibitory synapse I told you it's kind of axosomatic. So inhibitory synapses are made in the soma in this case, and the dendrite or the specialized structures is the excitatory. There's another cool thing. So the excitatory synapse, again, you see this PSD, the darkened portion, it's not dark here. The vesicles are larger of the glutamatergic synapses, and the vesicles are smaller here. Okay. But people have used these electron micrographs for years to really now reconstruct the different morphology or the cryoarchitecture of a cell. But these are some of the features that we kind of use to, even without any labeling, even without any, just looking into it in electron micrograph, right? And we see these different properties. Again, you have a lot of mitochondria. You can look into the changes of mitochondria. You have other structures as well. I didn't really comment on this SP. Can you guys think of this SP? I didn't comment on that. So it's fine. So it's extension of an endoplasmic reticulum that's coming into the spine. Oh, sorry, SR. Yeah, SR. All right. So actually, not really. It's the chemical nature of that. It is possible that even the vesicle of the glutamatergic vesicle could have different amounts of glutamate. So it's not the volume, but the type of chemical. So GABA is smaller than the glutamate, as far as I know, chemically speaking. Arco is thinking, am I correct? Maybe. But it's not the volume is what I know. So it's not that if you have a bigger vesicle, you'll have more release of neurotransmitter. That's usually not correlated. Anyways, so there are two synapses which we'll use as model to understand the different properties of synaptic transmission today. One are the synapses that are made on dendritic spines that people have used extensively to understand synaptic transmission. So this is example of a hippocampal neuron. This is CA1 neuron. It's a very beautiful structure. We also study that in the lab. And then if you just take one segment of the dendrite and zoom it in, you see this. So this is again a Golgi stain. And you see these thorny little projections. If you zoom it in, these thorny little projections are where the synapse is made with the other cells. So one connection, And if you basically do a reconstruction of that with the other neuron, you'll see that these sites where the contacts are made, this is where a synapse is made with the presynaptic neuron. So this CA3, CA1 synapse is used as a model synapse to study different properties of synaptic transmission. So we'll use that as our study model today. And then the other one is the frog neuromuscular junction that people use to study the synapses. So frog neuromuscular junction cartoon look like this. So it has these villi, which is where you have the postsynaptic receptors. In this case, these are acetylcholine receptors. What are the receptors in the CA3, CA1? Sorry, I didn't talk about that. Do you guys know what are the postsynaptic receptors? Why do you guys always start with NMDA? NMDA is like so not important for synaptic transmission. Remove that, erase that from your memory. A2, A, adenosine is there, but it's not a primary postsynaptic receptor. AMPA, yes, AMPA is a glutamate receptor. What other receptor? NMDA, don't worry about that. There's also a third type of glutamate receptor. It is important for your synaptic. mGluR does not participate in transmission. Again, gonna come for the second lecture. They'll all become relevant, but NMDA receptors are not important. mGluRs are not important. There's a third one that you guys don't really. Kainate receptors, absolutely. So there are these third type, which are kainate receptors. They are very similar to AMPA receptors, but they have very special properties of where they are on the synapse. But glutamate receptors, generally speaking, could be of these three categories: AMPA, NMDA, and kainate. And the kainate and AMPA plays in the basal synaptic transmission. So those are CA3, CA1. Here we have acetylcholine receptors. Again, acetylcholine receptors could be of two types, right? Which is the one that play a role in synaptic transmission? Nicotinic. There is nicotinic acetylcholine receptor and there is muscarinic acetylcholine receptor. Nicotinic acetylcholine receptor is the ionotropic, or the ones when the acetylcholine binds, it leads to ion influx. And when the muscarinic binds, or the muscarinic acetylcholine receptor gets activated, it's a GPCR. So it activates a G-coupled receptor signaling, or GQ or GS kind of signaling. So we'll look into some of the classical experiments that were done using this model system, where they had a frog neuromuscular junction, or this is called a frog muscle, or called as an end plate, and then how a motor neuron was making synapses onto these end plates. Okay, so we'll study that. All right, so the first experiment that I want to talk about are to understand how does the synaptic transmission or the synaptic vesicular release occurs. Scientists did a kind of cool experiment with the frog nerve stimulation. This is done by Heuser and Rees back in 1970s. So we already know quite a bit about action potentials now, right? This was back, action potential papers in 1950s and all. And here they looked into the end plate potential, where you see this kind of a structure of a villi here. And then the presynaptic thing, you have these synaptic vesicles. Okay? And they did simple experiments where they electrically stimulated this end plate for 0 minutes, 10 minutes, 15 minutes. And then after, for example, after 15 minutes, they observed that these synaptic vesicles are kind of depleted, suggesting that these vesicles are kind of getting fused and releasing some chemicals, which are bound by the postsynaptic part of the end plate. And they even, using some of these experiments, they then drew schematic of how this vesicular fusion might be happening, which is kind of like the budding of the synaptic vesicle cycle that we'll read about, how there are some vesicles which are a lot more readily releasable, and some vesicles which are in the reserve pool. So they started giving some of these terms like that. They also talked about a lot of other things, like the vesicles having nanodomains. So not all vesicles would respond to the stimulation in the same way. And we'll talk about how we've started to get some significance of that now. This is a very beautiful way of even they see how the recycling take place in a clathrin coated dependent manner. So they talk all about that just by looking at some of these micrographs. So this was their first paper that came out. And then after this, you guys should check it out actually. They also did another cool experiment where they wanted to get a temporal evidence of how fast this synaptic vesicle actually occurs. So they did another experiment or another series of experiment where now this is transmission electron microscope, not SEM, and they developed this cool thing where right, so you know how we freeze the slices and then we make thin sections of it. So in this case, to get the temporal information right before they would freeze the slice, they would actually stimulate them within, I would say, in a millisecond time scale. They would have yeah, the stimulating wires, and whereas yeah, so there is a copper cold block here, and they would have the slice here, and right before they freeze it with the help of a block, they would stimulate it. And this kind of technique is called as freeze fracture technique. Okay. So they would freeze it and then they would fracture the tissue into different parts such that, so what you see here is literally your presynaptic terminal from the bottom. So if you, so you're literally in the synaptic cleft and you're looking up into your presynaptic side, and you have these zones, again, absolutely active zones, where there will be vesicular release will happen. And you'll see that, let's see if I show. So there would be these dents. So these are the different phases. So again, they kind of theorized when what will be kind of the synaptic vesicle cycle. They will come, they will be primed, they will have exocytosis, and then they will go up. So then they found these different stages of synaptic vesicle cycle, where here there was a vesicle before, but now it has kind of been internalized. Here you have the release happening, so you can actually see the hole or the capacitance or the membrane getting bigger. And this is right after stimulation. So you can see the heterogeneity of the different stages of synaptic vesicle cycle on the same synapse, which was interesting. This doesn't happen in mammalian or in the central nervous system, but this is in the frog neuromuscular junction. But nonetheless, we use something very similar nowadays, but instead of doing electrical stimulation, we do something called as an optical stimulation, which is called as what is it called? Freeze fracture. Like it's flash and freeze. So it's flashing the light to activate your presynaptic axons that contain channel rhodopsin, and then you freeze this tissue. So you don't need this big trigger or stimulator. Okay, so far we have now started to understand how does the basic synaptic transmission take place, and these are some of the experiments that kind of started on explaining some of the phenomena. And now we know how it works. We'll just go into the different processes of that. Do you guys know about synaptic transmission? Should we all walk through it together? So what happens first? Action potential is formed at the, so I'll start. Action potential is formed and it propagates down to the axonal terminals. And then what happens? That's 3 steps. So it comes down and then it depolarizes that axonal terminal. What that does? So the voltage gated calcium channels open up and then there is calcium influx. What will that do? Some calcium sensor. Okay. Absolutely. So you have voltage gated calcium channels that open up, calcium comes in, there is some calcium sensor onto the synaptic vesicles that gets bound, and those vesicles are then, those are already primed, and they come and they do the exocytosis. So you have glutamate, or in this case, neurotransmitter that gets released. And then what happens? It's released where? The neurotransmitter? Synaptic left, and then bind to the receptors, and then the ion flow happens, right? So what do you think is the time duration of all this? It's in milliseconds. You see how quick an action potential. Action potential is probably what, 1 millisecond to 2 milliseconds, and then you have calcium influx that come in, and you only have a half a millisecond delay when you have a postsynaptic response. Isn't that like amazing how quick, even though we call it a slow mode of transmission, but it's only half a millisecond delay. That's insane, right? For me, that was kind of mind blowing how such complex synaptic vesicle signaling is happening so fast. Um, all right. Now, the one that I talked about is an example of a very specific form of transmission, which is an action potential dependent form of transmission. And that is, in today's term, it's called as a synchronous release of neurotransmitter. What does that mean? It means that when an action potential comes down, the neurotransmitters are kind of synced to that action potential. Okay? And again, this is based on the studies we did with our model synapses. But now we know that there are different modes of transmission. It's not just synchronous release. In this case, if you evoke or if you basically stimulate the presynaptic axon and generate an action potential, you will have an evoked response postsynaptically, which is now in this case, in a voltage clamp condition, it's called as an excitatory postsynaptic current. It's the response from the receptors. But you'll see in some of the example synapses, such as in thalamus, you have these asynchronous release of vesicles as well, which happens way after the action potentials come down. I'll not go into the detail of how does that happen, but it is to do with the calcium nanodomains and the delay in how the calcium diffusion happens in the presynaptic terminal. So there's an asynchronous release of events that you can see here leads to postsynaptic response in an asynaptic manner, asynchronous manner. In addition to the synchronous release of synaptic vesicles and asynchronous release of synaptic vesicles, you also have a third mode of transmission, which is action potential independent. And that is called as a spontaneous mode of transmission. So what would be the function of spontaneous transmission? The job of your neurotransmitter is to actually transmit information that's coming with the help of an action potential from the upstream, right? But why spontaneous transmission? Okay, why? Yeah, you see that? It's good you're reading. Uh, but what does that mean? What does that mean, homeostasis? How would that be maintained if you have a spontaneous release? So that regulates the release from these indirectly, kind of. Yeah, so it's not just within the synapse, it's also the nearby synapses. So if you have like an inhibitory synapse, so more increased inhibitory tone, to compensate for that, those inhibitory synapses activate the calcium influx through a different channel, which is not action potential dependent, and then can have release, that can have an increased spontaneous release. But more than that, if you don't have any mode of transfer, sorry, yeah. Yeah, it's not just sensing, but sensing postsynaptically. So if there's an increase in the chloride ions suddenly, right? So you can have increase inhibitory tone, and there could be a retrograde transmission that can control these spontaneous events. But more than that, maintaining an excitatory synapse is also important, right? So even if, for example, you have stored some information long time back, you've activated a connection, but you have not used it for a long time. You want to maintain those synapses. You might have learned how to ride a bike, but you might have not ridden a bike for like a long time. But how does the brain then keep those connections active? You're not riding a bike. Maybe I, like, for example, I have not ridden a bike in the last 10 years, but how does the brain really maintain those connections? And that is happening due to some of these spontaneous events. So it really helps in maintaining the synapses. It's one of the primary functions. If you don't use that, the things are going to degenerate. So the axon, it's not going to maintain that synapse. It's going to like either weaken, and then there'll be decrease in the synaptic density and the synaptic loss will occur. Um, or um, I mean that's so there are many ways that which will lead to synaptic loss. So it is going to conserve the energy if it thinks that that synapse is actually not important. It's a long-term thing. Um, yeah, it's a long-term information storage. Yeah, and so in a regulated manner, some of these spontaneous transmission mechanisms allow it to maintain those synapses. So this is one of the ways of maintaining some of that learned information. A lot of different ways. I think we discussed that, and we don't know for sure, but one of the ways we can do it is by having different signaling cascades that can participate in regulating this phenomena, right? If the signaling cascade, or if there is some change, for example, if it's very long-term information, there would be some structural change or some um, um, changes in the gene expression profiles that can have a lot more long-term regulation in a very different way than a basal synapse. Before your experience, before I open. Absolutely. So it's not just for learning. It's not just that once you have learned, you use it, but it's also used for maintaining the basal network or the connections that we need for even basic information processing. So it's the learning is one example that I give. It's kind of intuitive, but you're right. Even before that, for development purpose, we can have. And let me think if we have evidence for development. Um, because in the development it goes the other way. It's mostly that we have a lot more connections, and then we do something called a synaptic pruning. um, and so this is maybe in development, this is something which is kind of actively inhibited in the very young animals or humans. So this might get triggered when you want to maintain the synapses. We don't want to go undergo the synaptic pruning path. Absolutely. Yeah, and so this could play a role in that, definitely. Yeah. Um, lot of the things are different in terms of the proteins that are involved in these evoked transmissions versus, or synchronous and asynchronous release versus the spontaneous. A lot of the things are different, and even um, the pools of the vesicles that participate in these are different. The proteins that are involved are not. I mean, there are overlapping proteins, but you also have a separate set of protein that are only participating in these spontaneous release.
Comes into the synaptic compartment. Does it mean that the calcium sensors that are specialized vesicles that are made for spontaneous release, having different calcium sensors?
No, so synchronous and asynchronous are kind of together, but it has to do with, for example, we see something called as pair pulse facilitation, right? It's a short-term plasticity. In our case, it's still synchronous because it still happens, but the same calcium domain or residual calcium increase could happen or could lead to vesicles being released at different times instead of really all releasing at the same time. So another thing you should understand is this is coming from how many synapses? Many synapses at the same time. You're just evoking, but these small events are coming from different synapses at different times, right? So this asynchronous release, even though you have generated the action potentials, you really have these vesicular release from different synapses at different times, and that could be due to how the calcium is being diffused into the presynaptic terminal. But usually it doesn't happen. It happens during, if you give patterns of electrical activity, or if you give some stimulus which is different from your basal synaptic transmission. All right. Let's see if we go into this. We want to go into this. There are also different types of spontaneous synaptic transmission. One that can happen within the same synapse. So you have the vesicular pool in the same synapse, and then you have a calcium-dependent recycling pool. You can have, but they both go to the same active zone, or you can have an ectopic release, which can have receptors different. And then you have different synapses that take place in the same cell, which one is there to just spontaneously release or do the spontaneous transmission, and one which is a calcium dependent synchronous or asynchronous release. Now, let's come to the chemicals. So we know simple parts. We know that there are neurotransmitters, and then we also discussed about neuromodulators, right? So what are the differences between the two? Neurotransmitters and neuromodulators. Can neuromodulators be neurotransmitters and vice versa? So neurotransmitters are generally the chemicals that are participating in synaptic transmission. When I say synaptic transmission, what does it mean? That there is synaptic transmission taking place. What does that mean? Through the synapses, what is happening? The information is getting transferred. What does that mean? So it means that if you do synaptic transmission, the likelihood of the postsynaptic cell to fire an action potential increases, right? Or decreases, or I mean the potential change happen depending on the type of synapse. But there is some change in the potential. Does that, yeah, yeah. Does that happen with neuromodulator? Not necessarily, right? Neuromodulators not necessarily need to change your potential either towards threshold or away from the threshold. So transmission is usually increase in the ionic flux to change your potentials. So your transmitters, what are the major neurotransmitter peripherally? Peripherally meaning outside the brain, or when you have connections with the muscle. We just learned it's acetylcholine, right? So those are called as cholinergic synapses. And when we say it's glutamatergic, so depending on the type of neurotransmitter, you categorize them into different types of synapses. So glutamatergic is glutamate, GABAergic is GABA. What are the other synapses or neurotransmitter synapses that you can think of? Not necessarily in mammals. Which is that? What is it called? Okay. So it participates with neuromodulator or neurotransmitter. It's a neuromodulator, exactly. So there are serotonergic synapses in flies. There are cholinergic synapses, central synapses in the which are cholinergic in nature in flies in lower models. Anything else that can act as a neurotransmitter? Glycine, exactly. All right, so these are all neurotransmitters. So now you understand the difference between neurotransmitter and neuromodulator. Now, each of these neurotransmitters, such as acetylcholine, glutamate, GABA, they all can also act as a neuromodulator. How? Which is something that you guys pointed out in the beginning. You see how all of these neurotransmitters have an ionotropic receptor, but it also has a metabotropic receptor, right? You have nicotinic acetylcholine receptor and a muscarinic. So muscarinic is when the acetylcholine binds to the muscarinic, that's when it acts as a neuromodulator. It modulates the synaptic transmission. It does not participate itself, but it changes some aspects of the synaptic transmission. Same with glutamate. You have mGluRs or metabotropic glutamate receptors, which we'll discuss next time. The other classical ones that you guys have heard about, dopamine, of course, it acts as a neuromodulator. Um, Norepinephrine, and so neuromodulators are of different types. Can you guys think of um, as in I don't think you guys can think of. So there are neuromodulators that are synthesized somewhere else in some other brain region, and then they can go and affect the synapses in the other brain region. So these are called as an extrinsic neuromodulator. And the example of that is dopamine, right? Where is dopamine synthesized? Substantia nigra, ventral tegmental area. So the dopamine synthesized there, but it affects pretty much the entire brain. cerebral cortex, cerebellum, hippocampus, anything that you can think of, dopamine projections are pretty much every there. Another example is of locus ceruleus, which is where norepinephrine is made. Again, it's synthesized in this very small nucleus, but then it's projected pretty much everywhere. So those are examples of extrinsic neuromodulator. Okay? And then there are these intrinsic neuromodulators, which is synthesized locally and also released locally. What are the examples of that? One you see here, it's called substance P. So there are some of these peptides that act as local intrinsic neuromodulators. Could be one. So it can be released in autocrine fashion as well. BDNF is another one. Can be released and can act on the same cells. So any other example that you can think of? Local release, local synthesis, local release. Very important one. Come on. Nitric oxide, absolutely. It is not a neuromodulator. I mean, it can act as a neuromodulator, but nitric oxide doesn't have a a GPCR like receptor to bound. Exactly, presynaptic. So it does travel. Yeah, it's a questionable neuromodulator. Which other thing you guys know? Um, the endogenous ganja that we have, right? Endocannabinoid. Endocannabinoids are very major neuromodulators in higher order brain regions. Anything else? All right, so this is something that I can, these are the ones I can think on top of my head. But there are a lot of these, so but we broadly categorize them into these extrinsic and intrinsic neuromodulator. And so the one that you asked, volume transmission, it's mostly happening with the extrinsic neuromodulator, but there are a lot of now local and very synapse specific releases of intrinsic neuromodulator, or um, or even the extrinsic ones come and they can release in a very localized fashion. So the way they do the transmission, the way they do the effect on different brain regions could be very different. This is something that I'll share the slides. You guys can read about it. It also depends on where everything is being synthesized. So some of the small peptides could be synthesized in the soma, and then they get transported to the axon or wherever it's required with the help of motor proteins that we were talking about. Then we have amino acid transmitters that are processed right in the axonal terminal, because you need recycling of the thing where they have shorter lifetime. And then one important property is they are actively packaged into the vesicles with the help of transporters, and they require ATP. So it's a ATP dependent and a pH dependent phenomena to recycle or refill your neurotransmitters back into the synaptic vesicle. Let's go into the basics of how the synaptic vesicle, now with like series of experiments, some that I showed you, but series of experiments have now shown that this synaptic vesicle cycle is a very regulated phenomena, where it starts with refilling of the neurotransmitter, again in a pH dependent manner. Then it gets translocated and docked near to your vesicles, which is what we kind of saw, right? Those docked vesicles which were there. And then you have priming, which is again an ATP dependent phenomena. This all happens before action potentials comes in. So when action potential comes in, you have calcium influx, and these prime vesicles, there is a exocytosis. Now this exocytosis is also of many different types. It's not just there will be full fusion of the vesicle. There will be something called as kiss and run, where it basically just open partially and then you get endocytosed very quickly. But then there is endocytosis that takes place, and then it translocates back. So a lot of these vesicles which have released a neurotransmitter, they make something called as an endosome. And from there, the vesicles are formed again and filling happens. So now where are these neurotransmitters coming from to fill the vesicles? Are they synthesized every time? What happens? How they are collected back? Exactly. So they are reuptake, and which cell really helps to reuptake? Astrocytes, yeah. That's something that we usually ignore, but we've started to understand the importance of that. So the astrocytes are absolutely important in actively removing the glutamate or any neurotransmitter from the synaptic cleft, such that your receptors don't get desensitized, but it also then um, recycles it back to the presynaptic terminal. All right. See what we have next. Do you guys want to go into the detail of what exactly happens, or do you guys care? Or it's like, I'll just tell you briefly. We have some proteins that are membrane bound, such as SNAP21, syntaxin. The names are not important. And then we have some proteins like synaptotagmin and synaptobremin, which are bound to the vesicles. So when synaptotagmin senses the calcium, what will happen is there will be conformational change such that the synaptobrevin goes and binds to this SNAP21 and syntaxin complex. And this is where your the priming has happened, and then it goes to the fusion part. Okay, so synaptotagmin is one word you guys should, or one protein that you should think of as a calcium sensor that really senses this calcium influx, synaptotagmin. But again, there are hundreds of more proteins that are involved in really regulating these presynaptic vesicle machinery now. We already discussed about this, so I'll just skip. So I wanted to discuss, oh, are we almost out of time? Can I just discuss this last concept? Again, this is one of those classical experiments that where, which I think is going to answer some of your question that you asked. This is done by Bernard Katz. Like I said, he really loved recording action potentials, and he kind of was not in the favor of recording things in voltage clamps. So there is one very famous paper that he published after recording these spontaneous end plate potentials or changes in the potential due to presynaptic neurotransmitter release. He does a noise analysis of this in a current clamp condition to understand how does transmission takes place. And he was completely wrong because he does everything in the current clamp. And then the Anderson and Steven paper comes, which does the exact same thing in the voltage clamp conditions, and they were able to really identify the quantal nature of this neurotransmitter release. Nonetheless, this is the original study that actually started talking about the transmission release. So I'll discuss this. So in this study, what they were doing was they isolated a preparation such that there's an intact motor neuron and there is an end plate where they can study these neuromuscular junctions. And then they evoked or they electrically stimulated this axon and they recorded from the postsynaptic muscle end plate. Okay? Even sometimes in this active preparation, if they kept it in a physiological condition, even spontaneously, they would record these postsynaptic changes. So these are spontaneous or action potential independent forms of release that they would see. And then they would evoke it, and they can see like a much larger increase in the potential. And they wanted to understand or quantify this a little bit better. So what they did, actually, I'll just go into the experiment. So what they did, see if I need. So you have these spontaneous events, right? That happens. So what is happening every time this is happening? What is, so this is basic synaptic transmission that's happening, right? So you have a vesicular release, acetylcholine gets released and bounds to the acetylcholine receptor, and then you have a response. When they would do the electrical stimulation, this is all without stimulation. When they would do the electrical stimulation, they would have a much larger event like this. Okay, and they wanted to understand they wanted to quantify this, like what really is happening here, how many vesicles are there. They observed that these events are kind of quantal or linear. So either it's 0.5 milli, most of the time it's 0.5 millivolt, or sometimes 1 millivolt, 0.5 to 1 millivolt. Okay, and then this evoked response, what they did, they reduced the calcium concentration and they increased the magnesium concentration such that you have low calcium influx you have low vesicular release, and you have a lot of failures of synaptic transmission. So even when you'll evoke, they wanted to control. So they also reduced electrical stimulation. They wanted to control the response such that they get either failures or they get some successes, which is similar to your spontaneous. And they characterize this. Understood the difference? So this is spontaneous and this is evoked, but in the evoked, they saw this. Um, they evoked it in a artificial setting to kind of match the miniature. And then they looked into the amplitude of these events. And what they found was this increase is usually kind of quantal. So they calculated the amplitude of the end plate potential, and they just calculated the number of observations that they are seeing with evoked. And they saw that at 0 means these are a lot of failures. They saw a lot of failures with this lower electrical stimulation, higher lower calcium and higher magnesium. They saw this increase in the failure, but they saw kind of like a Poissonian distribution where a lot of the amplitudes were revolving around 0.4. Now it's not exactly 0.4 because there are a lot of these technical challenges to really calculate the absolute voltage. So they had this range between 0.3 to 0.5 or 0.6. But there is an increased observation at 0.4. Again, there is an increased observation at 0.8. What does that suggest? That there are two vesicles released. Now the change in the potential is almost double than the first. Then you have again 1.2. Now there are three vesicles released, and then you keep going like that. And these are some of the classical experiments which suggested that the release of neurotransmitters is quantal in nature. It's not that you'll have a continuum of the potential responses. You'll either have, it's going to jump from 0.4 to 0.8 to 1.2 and so on. And this is one of the important properties that we use to um, study synaptic transmission. So there are three. Now this quantal nature gave us, or studying these properties of synaptic transmission gave us three important features, which I'll end with. The three features, which is what will be the basis of our experiment next on Thursday. The three features are: one is N, P, and Q or I. So N is the number of vesicles that are there in the synapse. That determines your synaptic, so that you can use that to define your synapse. The probability of release of these neurotransmitters. Sometimes you can have really high probability of release. Sometimes you'll not have as high. So you have low probability of release. So it's not that every time an action potential comes down, you'll have vesicular release. So yeah. So for example, hippocampal synapses, every 5 times an action potential comes down, you'll have a vesicular release. So those are low fidelity synapses. And then the last one is the Q or I. So the quantal content that gets released from the presynaptic terminal, and then the single synapse current that you see, or the potential difference that you see, is your I. So those are three properties of the synapse that we'll use to see how, when we change this, we can actually process information. Okay? And I'll just end with this example of high fidelity versus low fidelity and the function of that, right? So for neuromuscular junction, those are high fidelity synapses. The probability of release is always 1. Every time the action potential comes down, you will have a acetylcholine release, and you will have receptors bound, and you'll have a muscle response, muscle twitch. This is very important physiologically speaking, right? The fidelity needs to be high if you want to have communication with the muscle. You want to have the motor movements. You want to have response to any kind of sensory stimulus. You need very high fidelity. Whereas in the central nervous system, such as hippocampus, the fidelity is very low. You don't really respond to all types of information that comes in. Okay, so I'll end with that. Yeah, so with the synaptic strengths, the P depends on, again, so different aspects of the vesicular cycle. The number of readily releasable pools, how many of them could be part of the active zone of where the vesicles are primed, and all that. the postsynaptic, you mean, or the? Mm-hmm. Yeah, it's the same thing. So yeah, absolutely. No, that's how we think about it, right? So the NPQ are not so they are going to define the synaptic strength, but we can also use that to study the synaptic properties, right? So you're right. So underlying P could be a lot of that back-end synaptic vesicular structures or cycle that determines P. The number of vesicles are again a property of the synapse. So yeah, absolutely. Cool. Any other questions? Some of them could be dependent on each other. But contal content, for example, is independent. The contal content, for example, is independent. I think N is also, I think these are three exclusive as far as I know. There are three independent events. What's, yeah, total number of vesicles are different than the proportion of the different proportion of the vesicles that determines the probability. But I think these are three independent properties. Probability of release and the amount of vesicle that gets released is not the same. Would it be mechanistically linked? Probability of releasing is happening is either 0 or 1, but when 1 happens, some of them will release larger contal vesicles versus less conta, right? So it's either yes or no for the P, but if yes happens, then how much yes is Q. The probability of is the probability of how many vesicles are going to get released, or whether the vesicle is going to get released when an action potential comes down. The quanta is more of from an individual vesicle, how much neurotransmitter gets released.
So how do you like calculate that get to identify the number that's on the active side?
Usually, a lot of these parameters are calculated by recording postsynaptically. So the P is more of whether you see a response versus you don't see a response when you have a bare minimum stimulation. So for example, if your probability is increased, you will not have as many failures. You will have much reduced failures. Yeah. So I skipped a slide. So there is a way to presynaptically where if you can just patch the presynapse, you can measure the capacitance. So for example, if you have a synaptic vesicle exocytosis that's happening, you have an increase in the capacitance that you can actually record or quantify. So even that happens if you record the capacitance, that happens in a stepwise manner. If you have one vesicle release versus two versus three. Cool. Any other questions? All right. Cool. So we'll continue on how now these transmission properties change in the next class, which is basically the underlying mechanism of synaptic plasticity, and then what's the consequence of that. Cool. All right. Thank you.